Okay, thank you. So we have started our discussion for seed science and technology with a topic of floral biology and seed physiology, in which we have covered all the basic aspects which are very important for your exam. So one last topic in the seed physiology is storage reserves. So few of the things are overlapping here because we have already discussed while discussing the seed development, since the storage reserves play a very important role during the development. So I have told you about the different storage reserves that are being accumulated during the seed development. So now what we can, or what how we will continue is, we will see like how these storage reserves are actually synthesized in the seed or how they are transferred, and then after transferring and seed filling, how they are utilized by the seed during the development, how the actual breakdown of these storage reserves happens, and also what are the different enzymes which will help in the breakdown or mobilization of this seed storage reserves. Okay, so this is what we are going to discuss today, and it would be very short session since we have only left out with the single topic. So also we will talk about the germination process. I have few things to show you or discuss with you. We'll do that, okay, along with this seed storage resource. So starting with, can you just tell me like what are the different seed storage reserves which we have discussed in the earlier classes, or the biomolecules? They are carbohydrates, yes, lipids, proteins, and lastly we have discussed about phytins. So we'll just leave these phytins aside and we will discuss about the carbohydrates, lipids, and proteins. So out of these lipids, which is the major storage carbohydrate? It is the starch, which is in the two different forms. So it is amylose and amylopectin. This is alpha 1,4 linkage, and this is alpha 1,6 and 1,4 linkage. So these have the monomer units of glucose molecules, both amylose and amylopectin. And the amylopectin is a branch chain or branch structure, and amylose is the linear structure. I hope you people remember all these things which we have discussed in the previous class. Yes. So now what we can discuss is, we'll just discuss the biosynthesis of this storage reserves. We'll start with carbohydrates, specifically starch, and then we'll move to the proteins and then lipids. So biosynthesis of starch. So what is the source of starch for the plants? Like how the starch is synthesized in the plant? So I think we have discussed about the relationship between the source and sink, right? Source is the part of leaves which acts as a source because this is the place, or this is the region where the glucose is synthesized by the process of photosynthesis. Once the glucose is synthesized, it is converted into sucrose, and this sucrose will be transported into the developing seeds and it will be accumulated there. So this is about the biosynthesis of starch. And the starch composition we have discussed, it is of amylose and amylopectin. And about the synthesis, this synthesis usually occurs in the daytime, but the mobilization will be in the night time. So what is the difference, mobilization and synthesis? Synthesis is a formation, mobilization is a breakdown. The breakdown of this starch usually occurs in the night, and its synthesis takes place in the daytime. And this is by the plastids in the leaves. Organs are the plastids where the photosynthesis takes place. You already know it, right? So this is about the starch, and we have one important thing to discuss here is about staining. So how to detect whether the starch is amylose or amylopectin? It is by the help of the staining, right? we have certain staining procedure where we will be able to differentiate between the amylose and the amylopectin. Can anyone tell me like what is the stain and what is the procedure? See, whenever this starch is treated with the iodine or dipped in the iodine solution, the color of this starch changes. So it is unique for amylose and amylopectin. So when you stain this amylic or add this iodine solution, so if the starch is amylose in the first place, it will stay a blue color. When the amylase content or the starch content is exposed to the iodine, if it gives the blue color, it means that the content is amylose. But when this starch gives us the red color, when it is exposed to the iodine, then it means that the starch content is mainly of amylopectin. So this can be the very important thing for you, because it might be, they might ask you the things like staining of starch with the iodine gives blue color, so what is the content in it, like this type of questions might be asked in the exam. So just remember it, when it is treated with the iodine, if it gives the blue color, it is mainly because of amylose, and if it gives the red color, it is because of amylopectin. So when this photosynthetic assimilation takes place, so photosynthetic assimilation with the help of carbon dioxide, when this occurs, the output will be either starch or sucrose. That is, the utilization of the CO2 will ultimately give you either starch or sucrose, which are very much needed for the seed development. And once they are produced, and they will be transferred into the seeds. So this is about the starch synthesis. Just a brief about these things, because in detail you need not to read about like how the process is occurring and what are the different steps of how it is being taking place. But when you study this crop physiology, it's very important for you to understand this photosynthesis, the complete process of photosynthesis, whether it is light reaction, dark reaction, the ATP that are being consumed, and the product that we are getting out of the in the light day reaction and the dark reaction, and what is the difference between the C3 plants and C4 plants and CAM plants. So in which of these plants the photorespiration occurs, all of these things you should be able to answer. Okay, so make sure to study this process of photosynthesis in detail while you're going through the crop physiology subject. Not only photosynthesis and respiration. There are three main important topics that you should cover from crop physiology, that is respiration, photosynthesis, and also the nitrate assimilation. This will be also included in the microbiology, so few of the content will overlap, so that is ultimately benefiting you. So make sure to cover all these things. And in the respiration, the number of ATPs that are generated, that is the most important thing, and number of ATPs that are consumed in case of photosynthesis. So make sure to remember all these things. Rather than just remembering it, just go through the complete process so that you will have the better idea about it. If in case they ask you any applied questions based on these two process or reactions, you will be able to answer them. I have talked till the production of sucrose. So now we have a sucrose. So which line I should repeat? Is there any disturbance from my end? Can you please answer me like others? Okay. Okay. So I was saying that the photosynthetic assimilation of CO2 will yield ultimately the starch or sucrose. So for this, to understand this complete process, you need to go through the photosynthesis topic, right? So while you cover this photosynthesis in the crop physiology, other two topics are also very important, which are respiration and nitrogen assimilation or biological nitrogen fixation. So in case of this photosynthesis, you need to have a clear-cut understanding about the difference in the C3 plants, how the process of photosynthesis is occurring, and the C4 plants, how it is different from C3. There are some bundle sheath cells that are involved in case of C4 plants, and also how the process of uh, photosynthesis is difference in the CAM plants. So how the water uptake is less and how they are more efficient. So all these things you should be able to answer, and also the number of ATPs that are being utilized in the different process. So when I talk about Kelvin cycle and light reaction, dark reaction, what are the different products and the reactants that are involved. So all these things, and they might ask you any type of question among these type of process. So make sure to not only to remember, just make sure to read it once to understand the process completely, starting from the scratch, so that you will be able to better remember it. So even after understanding the concepts, it is must and should that you should be revising it from time to time. But if you don't do it, like you will be forgetting it. So even me, like it's been a year that I have not gone through this photosynthesis, so I'm not able to reproduce the same for you all. So this is how it goes. Okay, so make sure to revise them from time to time. So major confusion will take place in case of ATP generation and the different reactants and the products that are formed, and different steps and stages. For example, in the respiration, it all starts with glycolysis, and next is Krebs cycle, and third one is oxidative phosphorylation, and fourth one is electron transport chain. So just remember the sequence, and this glycolysis is in both aerobic and non-aerobic. Rest of the process are aerobic, and this occurs in the cytoplasm. The glycolysis occurs in the cytoplasm, and this is in mitochondria. So the organelle which is actually involved, and what is the actual process going on. So these are some basic things that you should be able to answer. If they ask you or not, it's a secondary thing, but these, since they are very basic, you should be having a clear cut idea about all these things. So coming back to the discussion, what I was discussing is, I was telling you about, we talked till sucrose. So now we have a synthesized sucrose that is ready to transported into the seeds. So how this transport occurs? I hope like you have heard about two different pathways, that is apoplast and symplast. Can you tell me the difference between apoplast pathway and symplast pathway? So the transportation of the sucrose usually takes place by two different pathways. So one is apoplast pathway and second one is symplast pathway. Can you differentiate between them? What is the actual difference between apoplast and symplast? So when I talk of apoplast, it is directly through the cells by the cell membrane, or sorry, by the cell wall invertase. This is the enzyme which is mainly responsible for the transport of sucrose through the cells. So when we talk of symplast, it is mainly through the plasmodesmata, that is cytoplasmic connections, and this is by the enzymes, the sucrose synthase. So if you just see the two different pathways side by side, so you'll get to see the difference. So here, yeah, through the plasmodesmatal connections, it is symplast. Yes. So if you just see the two symplast and apoplast pathways side by side, you will get to see the difference. So in the sucrose, this is sucrose synthase here, and it is cell wall invertase, which is actually involved in the transport. So it is, in the first case, it is converted into UDP glucose plus fructose, and this is because of other, like by the help of other enzymes, this is finally converted into glucose 1 phosphate. Whether it is symplast or apoplast, the final one is glucose 1 phosphate. Why? Because at the end of this apoplast pathway, once this glucose 6 phosphate is produced by the help of phosphoglucomutase, so it will be again converted into glucose 1 phosphate. So after the formation of this glucose 1 phosphate, next we have a same steps for both 1 and 2. That is for both apoplast and symplast pathway. And here it goes like glucose 1 phosphate is converted into ADP glucose, and it is by the enzymes called as granule bound soluble synthase 1 and 2. So this will convert this glucose into amylose, and from this amylose, if the one is involving, it is for storage tissues. If in this case, while conversion of this ADP glucose to amylose, if the granule bound soluble synthase is soluble synthesis 1 is used, then it is for storage tissue, it will go and store. But if the granule 1 soluble synthesis is used, 2 is used, it is for leaves and non-storage tissues. That is, this amylose is transported to non-storage and leaves. So this is about the amylose. So in case of amylopectin, this ADP glucose, with the help of the other class of enzymes which are soluble starch synthase. So this will involve and this will branch the glucose molecule and the amylopectin are formed. Again, there are several other enzymes here. One main enzyme is AGPase, which is also called as ADP-GPase. So this is the heteromeric which consists of 2 alpha and beta chains. So you need not to worry about all these things, but at least remember what are the different enzymes that are involved. So mainly it is because of cell wall invertase and sucrose synthase. That is the mainly involved in the first step of breakdown of this sucrose. So this is in case of sucrose synthesis in case of symplast pathway, and in the apoplast it is cell wall invertase. So this is the main difference between how the apoplast pathway goes and how the symplast pathway goes. So one is through the cells and one is because of the cytoplasmic connections, which is plasmodesmata. I hope this is clear. Just confirm me once with a yes if it is clear. I'm waiting for your answers. Okay, yes. So the protein synthesis, I hope there is no need to discuss the protein synthesis here since we have already done it in the genetics class, right? So nextly, it is lipid synthesis. It is a bit complicated and not even for your syllabus. So just skip it. You don't need to worry about how this lipid synthesis actually occurs. So you can just skip this lipid synthesis, but at least for a basic understanding, you at least have to know how the starch synthesis takes place. So rest is protein synthesis. I hope you already know how the protein synthesis occurs. So this last one, lipid, you can just skip it. So this is the overview of just seed storage results. I have not spoke about the mobilization yet. We'll skip go to the mobilization once we discuss this germination process again. I have skipped few things in between, so I've added these things here. So we'll discuss this and then we'll move forward. So germination. So what do you understand by germination? There are two different, there will be a different meaning when we talk in terms of different field or the different area. For example, according to seed physiology, so germination is the protrusion of radicle. Just the protrusion of radicle. If there is any outgrowth of the radicle or if it breaks open the seed coat, so then we immediately call it the process as germination. So this is in case of seed physiology. In the perspective of seed physiology, just a protrusion of a radicle is termed as germination. But if we are doing seed testing, if we want to know the germination percentage, so it's not only the protrusion of the radicle, it is the complete emergence of a seedling. It is complete emergence of a proper seedling from the seed. Only after that we will consider that this particular seed is germinated. So this is how it is different from the seed physiological point of view. So seed physiology is nothing but a process where if there is a protrusion of radicle or it is break opening the seed coat and coming out, the structure is actually coming out, it means that the germination process has been begun. But in case of seed testing, so it's all about vigor, whether there is an ability to germinate or not. It's about the ability. So when we are assessing the ability, we cannot directly say that protrusion of radicle can be of a germination, because after the protrusion of radicle, though it sometimes it won't develop into a complete seedling, or it cannot give rise to a complete plant. So that will be seen only after a few days. So we will wait for a few more days, and when the proper seedlings has been come out, then we will distinguish it as a germinated one. So this is how it is different from seed physiology. So what are the different factors that are affecting the germination? These factors, I think, they are very common to various physiological process. like it's again the water, temperature, light, and the nutrients which we have discussed till now, and the oxygen. So these are the important factors which are responsible for the germination, or these are the important factors which affect the germination process. So what about the difference between the types of germination? So I have just told you the germination now, how it occurs, the process of germination. So what are the different types of germination then? So there is this one important thing which you should be remembering this every time, that is hypogeal and epigeal. Yes, very good. So hypogeal, epigeal germination. So I have a figure for you. You can just see the difference of what is hypogeal and what is epigeal. So if you just understand the different structure here, so let us start go with one by one, where we will discuss this hypogeal first. So in case of hypogeal, this is the radicle and this is the cotyledons, or this is the shoot part and this is the root part. Just erase this again. Okay, this is the root part and this is the shoot and this is the seed. So when you just sow the seed in the soil, you see the protrusion of radicle first. Okay, roots are formed, fine. But when you see the epicotyle, you will see the cotyledons will remain below the soil. The below the cotyledon, we know that it is hypocotyle. So the hypocotyle is the region between the root and the cotyledon, and the epicotyle is the region between the shoot and the cotyledon. So in case of hypogeal germination, the cotyledons will remain below the soil, and it is only the epicotyle which will elongate, or which will come, like emerge out of the soil or come above the surface. So this is called as hypogeal. So the hypocotyle cotyledon will remain below the soil. It is only the epicotyle which is elongating, which will come to the surface. So this is hypogeal. So when we talk of epigeal, so this is again the radicle and the seed, but the difference is, instead of epicotyle, this is a hypocotyle which is elongating. From the roots are formed. Okay, fine. It is same for both epigeal and hypogeal, but after the formation of roots, the hypocotyle actually breaks open and come above the soil surface. So we can actually see the hypocotyle in case of epigeal germination. So this is the hypocotyle, and above the hypocotyle, the cotyledons are well above the soil surface. After this soil surface, it is the region between the shoot and the cotyledon that we observe the epicotyle. So when we just talk about the definition of epigeal germination, see, the hypocotyle will elongate and drags this cotyledon out of the soil surface and comes above the ground. So this is epigeal. So when we, in case of hypogeal germination, it is the cotyledon which will remains below the soil, and it is only the epicotyle which will emerge out of the soil surface. So this is the difference between epicotyle and hypocotyle. So what are the examples then? Examples for hypogeal and epigeal. And what about the groundnut? Whether I should place this groundnut in hypogeal or epigeal? So I hope like many of you will be very confused with this particular groundnut case, since in few books they have mentioned it as epigeal, and few of the books it is hypogeal. When you just Google it, it will show you that groundnut is epigeal again. So can you tell me like whether this groundnut crop is epigeal or hypogeal? Yes, like you are still confused, or you don't want to answer. You feel it is very simple, like you already know it. Okay, see, already there is one answer that it is epigeal. Okay, see, I'll just tell you the one simple trick. In case of grams, peas, beans, exception is broad bean and runner bean. All other beans will be usually epigeal germination. But in case of grams and peas and cereals, it is hypogeal. And also the red gram, which is again the gram, this is also a hypogeal type of germination. So talking about epigeal, these are the examples. So this is how you can remember it. You need not to just remember it for all the crops. These are, since they are major pulses and cereals, so and also few vegetables, so they are given it. So you just remember these examples. So any one of these questions will be asked in the exam, or this example will be asked in the exam. Okay. So about groundnut, I should answer this, or you will be answering this question whether it is hypo or epigl. Yes, Nanika, you want to answer? Yes, answer me. I don't think like you will be able to unmute. I don't know why. So you can just type in the chat box if you are not able to unmute yourself. Okay. So groundnut, just instead of just falling into the controversy with your friends or into the debate with your friends, one simple thing that you can do at your own place is just try the germination test. Will you do it? Can you just do the germination test of the groundnut that you will get? Even the groundnut that you use for that is which is edible, even that will germinate. So just sow some groundnut seeds and see, like, after, might be its germination time is around 3 to 4 days. Radical protection will takes place within 4 or 5 days. Just observe it. If you do it yourself, it will be better. You will be remembering it better. Okay, you won't forget it. Just do it once. Anyhow, I'll be mentioning in my notes. But if you are a bit excited about knowing like whether it is hypogel or epigel, or you want to really look into the structures of the groundnut, then you can just do a germination test and look by yourself. Not actually the groundnut only. Like, even if you have a doubt on any other crops, just do the germination test and you will be able to answer yourself. That would be the best idea, because it only takes 4 to 5 days or maximum 7 days for germination to occur. Yeah. Okay. Very nice. So Avik has done the germination test. Fine. Very good. So, and this is hypogel. But there are few students and few seniors, few juniors, few friends who will still debate it as hypogel. But if you want to clear by yourself, you can go through the germination test, because we have done the germination test by ourselves. I will also share the picture of it later. Might be tomorrow. I will share with you, and it will be posted in Instagram as well. You can observe there. So if you are just wanna observe it with your naked eyes, just try it out. It's not a complicated process to do. You just need to sow it once. Okay. So why I will tell you like why there is a confusion even after germination test. Why? Because sometimes the cotyledons will actually split into two halves. One half will be below the soil and one cotyledon will be the above the soil. So in this case, since this is occurring, many of them will think that okay, cotyledons has come out of the soil surface. This is epigel, not hypogel. So, but few people will argue that since the complete cotyledon has not come up to the surface, it is epigel. But actually, when we call the germination as a hypogel germination, so the hypocotyl should elongate above the surface. The complete cotyledon should be above the surface, and the below the cotyledon, the surface or the region which is called as hypocotyl, which should be above the surface. If it is not above the surface, then obviously this is a hypogel type of germination, not epigel. Okay. So I hope I am very clear with you all. Yes, just give me confirmation if it is clear. Okay. So we have another type of classification for germination, that is based on the light requirement. I hope like you have heard about the terms called as positive photoblastic and negative photoblastic. So what is the difference? Positive photoblastic and negative photoblastic. So positive photoblastic is the type where it requires the light, or the crops are categorized into the positive photoblastic, which requires the light for the germination. The major examples are lettuce and tobacco. And the negative photoblastic, they do not require light at all for the germination. So the onion, lily, amaranthus, and nigella. So these are some specific example of negative photoplastic crops. And I have just given some important note here. I don't know whether you'll be able to understand all these things or not, but I have given it. Why? Because you can expect these questions from in your exams, but to understand in these all these things in detail, you need to just dive deep into this C technology, which is not at all worthy. So I just mentioned it as an important note. Just read them once. So this first thing you will be able to understand it. Why? Because the hard seed we have discussed. If I say that the seed is hard, it means that the structure or the seed coat is impermeable to the water. So if this is the case, there can be no imbibition. So that is why in case of hard seeds, we don't see the imbibition directly. That is why the hard seeds cannot germinate in the field condition. So they are given a seed treatment where they will be soaked for 48 to 24 hours before the actual sowing. So if they are pre-soaked, the structure will be little spongy, and then this imbibition process can take place. So, and the next one is about the dead and dormant seeds, the phase 1 and 2. The phase 3 doesn't occur here. So there are total of 3 phases of germination, out of which only phase 1 and 2 will be seen in this dormant and dead seeds. The phase 3 we don't see it. So the reason why it occurs, don't I need not to explain it, like, because it is not required. Just leave it, okay? But at least remember, if they ask, in case if they ask, just remember this thing. So, and again, the imbibition, I told you, it's just a mechanical process where if you just place the seed in the soil, because of the difference in the osmotic potential, there will be exchange of, or there will be a flow of water. Okay, I'll just tell you the one interesting thing here, or interesting process of how you can understand the germination, the process of germination. See, the understanding the process of germination is very simple. For example, if you just sow the seeds in the soil, so what are the different steps that occurs when you just sow the seeds in a soil? So the embryo inside the seed will absorb the water. This is the first step. In the first step, what happens? The embryo will absorb the water and it will start swelling. Since the water content in the seed will increase, the embryo will start swelling. So once it starts swelling, it will produce some signals to the hormones, or it will synthesize the hormones, which is mainly, which is the hormone? It is GA3, that is gibberellic acid. So this will activate this gibberellic acid. Upon activation, this gibberellic acid is transferred or diffused into the aileron layer. If this is the seed and this is the embryo, and the aileron layer is at the endosperm, outer part of endosperm. So from this embryo, the GA3 will diffuse into the aileron layer. So what are the important class of enzymes that are present in the seed coat or aileron layer? If you can remember. So they are nothing but hydrolytic enzymes. So the hydrolytic enzymes are mainly present in this aileron layer or seed coat. So because of transportation or because of arrival of this gibberellic acid, which is the hormone, it will activate this hydrolytic enzymes, or it will signal the synthesis of these hydrolytic enzymes. Once these hydrolytic enzymes are synthesized in the aileron layer, it will again diffuse into the endosperm. Why endosperm? Endosperm is the main storage tissue. So once these hydrolytic enzymes are passed on into the endospermal tissues, in the endosperm, the storage resource breakdown will takes place. How breakdown? This is why they are called as hydrolytic. The process of hydrolysis will take place. That is, the complex product, like complex molecules or compounds, are breakdown into a simple molecules, after which the seed can actually utilize it. So this is how the enzyme breakdown will occur, and the storage reserves mobilization will takes place, and as a result of which the radical protrusion occurs. So this is a simple process of germination, how actually it takes place in the cell or inside in the seed when you just sow it in the soil. It is all because of the water. The water triggers the synthesis of a GA3, and upon which the GA3 will triggers the synthesis of the hydrolytic enzymes, and these hydrolytic enzymes will move to the endosperm. And in the endosperm, since it is a storage tissue, and in the dicots, cotyledon is the storage tissue. Upon transport to these structures, it will mobilize, or the enzyme breakdown will take place, and all this breakdown and nutrients will be utilized by the embryo to protrude the radical, by which the seed coat will break open and the structures will rise or come out of these seeds. So this is about the germination. I also provided with the overview. You can just go through this overview once. So when I talk about the overview, it is mainly the process of germination. After germination, the respiration takes place. Seed respiration starts. After respiration, it is RNA and protein synthesis. After synthesis, it is the complete germination of or the completion of the germination. So these are the four major stages of the germination. How the germination occurs in the seeds. So again, the individual stages are differentiated into different types. For example, the imbibition I told you earlier, it is a triphasic one, the phase 1, phase 2 and phase 3. So these are the triphasic case of imbibition. And next, the respiration, it can be aerobic respiration and also the not anaerobic, it's absolutely aerobic. And in case of aerobic, you already know the process of aerobic respiration, the different stages. So the same process occurs here, and then there will be RNA and the protein synthesis, and it is after that completion of germination. So this is how it takes place. This is anaerobic and aerobic. Anaerobic is restricted to only glycolysis, which will give you the ethanol or lactic acid. I think I just okay. So there are some special structure for uptake of the water that are very specific to certain crops. For example, it is the chalazal cap in case of Cystaraceae family and Malvaceae, and the strophiol plug in case of Papilionaceae, and in the case of Musaceae, it's operculum. I hope like you have heard about the term operculum, right? It's nothing but the integments that is present in the seed coat, which is derived from the micropyle. So there are some special structure which have been evolved. So they have a function of uptake of water, and they're limited or they are specific to certain crops. Okay, this is the overview of germination. So next we can start our discussion with the mobilization event. So once the proteins are mobilized or proteins are broken down, what is the end product that we get? And if the starch is broken down, what is the end product? And what about lipids and the phytin? At least tell me about the proteins and starch and lipids. What are the monomeric compounds or molecules that you get from the breakdown of these polymers? Yes, it is nothing but amino acids. Why it is taking too long? Yes, amino acids, glucose, and this is fatty acid plus glycerol. I told you like lipids are made up of fatty acids and glycerol, right? So this is phosphorus, calcium, magnesium, and also the inositol. Yes. So these are the monomeric units of the breakdown of all these complex structures. So once this breakdown occurs, all will be converted into the glucose. Since the glucose is the primary source for the utilization in the seeds, so this will be glucose will be utilized by the seeds. So this is how the mobilization event occurs. And most importantly, what you should remember here is the enzymes which are involved in this breakdown. What what are the different enzymes that are involved? So when I talk of proteins, it's nothing but the proteases. It's not that simple that the common name is proteinases. There are several classes of proteinases which are responsible, but broadly it is proteinases. And in the starch, it is amylases, and the lipids by the help of lipase, and phytins it is phytase. So this is a simple representation of how during respiration the seed storage mobilization will takes place. So in detail, I have just mentioned about the starch, I think, how the starch degradation takes place. So when I talk of monocots and dicots, what is the difference and where is the difference between the starch degradation? Yes, of course, like starch will be present in both monocots and the dicots. Like how it is different, like whether the process of degradation is different from monocots and dicots or it is same. Actually, it is different because of one major enzymes where dicots usually involves phosphor phosphorylases which are present in the cotyledons, which will break down the first step, and in the monocots it is the alpha amylase. So in case of monocots, it is the alpha amylase which will break down both amylose and amylopectin, and after which it is again the glucose 1 phosphate, and ultimately it is the sucrose, which is formed. So this is in case of monocots. So when I talk of dicots, it is mainly because of starch phosphorylase, which will first convert this amylose and amylopectin in the glucose 1 phosphate and again the limit dextrin. And again, the rest of the process are very similar to the monocots. So once the glucose 1 phosphate is formed here, it is similar to the monocots, where with the help of UGPS, it will be converted into UDP glucose, and again it will add up with the fructose to give the sucrose. So this is how it is mobilized. This is for starch. I think I have not mentioned it about proteins because it is ultimately same. The protein degradation is because of again for the monocots, it is because of the carboxypeptidases and aminopeptidases. As I told you earlier, there are complex, like there are several categories of the proteinases which are actually responsible for the protein degradation. So which have some specific nature where they will cleave. For example, the carboxypeptidases will cleave the terminal amino acid from the carboxylic end. That is why the name carboxypeptidases. Aminopeptidases will cleave the terminal amino acid from the amino end, and they are nothing but they are broadly called as endopeptidases, which will cleave the internal peptide bond, and the oligopeptides are formed. And after the once these oligopeptides are formed, but again by the hydrolysis, you will get to see the amino acids. So this is how it occurs in case of protein degradation. So how it is again, how it is different from the dicots, how the monocot protein degradation is different from the dicots. So you see the storage proteins is cleaved by the help of carboxypeptidases and aminopeptidases, right? See, this is a single step process which is directly converting into amino acids, which I have elaborated here and I've showed you how it actually occurs. Okay, don't get confused with that. So in case of dicots, these proteins are usually stored in a special bodies which are called as protein bodies, and this is ultimately in the cotyledon, not in the endosperm in case of dicots. So the storage proteins, with the help of these three major classes of enzymes, it will give us the amino acid. And this amino acid is transported in the cytosol and then to the target site. So targeted sites. So this is how the mobilization occurs in case of the protein. So these proteolytic enzymes, the enzymes which I mentioned here, where they are synthesized. So just remember this point, it's very important. The proteolytic enzymes are synthesized in the endoplasmic reticulum. Okay. So next it is lipids. I hope like I'm going too fast. Why I'm going too fast is you need not to invest so much of time in understanding all these things, but you should be able to remember the things at least step by step, how it is occurring, why it is occurring. Don't see it elaborately, but how it is occurring and what are the different enzymes involved, that can be important. Just remember those things. So next is lipid mobilization. So the oils are generally stored in the oleosomes, which are made up of three different proteins. The oleosomes are made up of three different proteins, which are oleosin, caseosin, and stereolysin. So out of these three different proteins, the oleosin is most abundant here, which forms the major component of these oleosomes. And in the most cases of the lipids are actually stored in the form of triacyl glycerides, and these are produced in again in the endoplasmic reticulum. And this TAG or triacyl glycerides will act as a major source of energy during the seed germination. So what are the functions of these three different proteins? I mentioned it here. You can just go through it once. Okay. So again, it is TAG, which is the main source of energy for the seed germination. So about the mobilization, okay, I think I missed something. I missed the process of lipid. Okay, just a minute. I have not just included the last slide here, which is the lipid synthesis. I will just mention the same. See, one, the lipid mobilization I was talking about. The lipid mobilization is from the triacyl glycerides. These triacyl glycerides are first converted into fatty acids plus glycerol, which is lipids. The lipids are first converted into fatty acids and glycerol, and this fatty acid is converted into acetyl CoA. So this fatty acid is converted into acetyl CoA, and it is because of enzyme which is fatty acid CoA synthase. So the enzyme which is responsible for the conversion of this fatty acid into acetyl CoA is called as fatty acid CoA synthesis. So this is a very important and very common reaction. Can you just name the reaction of conversion of fatty acid into acetyl CoA? I believe like you will be very familiar with this reaction. It is called as beta oxidation. So I believe like you have seen in several papers, they will ask you regarding name the organ where this beta oxidation takes place, or name the part of the plant where the beta oxidation takes place. It is in the peroxisomes. So just remember this point. The beta oxidation is nothing but the conversion of the fatty acid into the acetyl CoA. Again, the process is it's lengthy. It again consists of 4 different stages, but at least remember the reactants and the products that you are getting from the beta oxidation and where it is taking place. It is in peroxisomes. Once this acetyl CoA is formed, then this acetyl CoA is converted into succinate. So this particular cycle, it is by the cycle called as glyoxylate cycle. After beta oxidation, it is because of glyoxylate cycle, the acetyl CoA will be converted into succinate, and this is in the glyoxysomes. Once the succinate is formed, we'll get the malate, and this is because of partial Krebs cycle. If you just observe the Krebs cycle, because of the partial Krebs cycle, there will be conversion of the succinate into the malate, and this occurs again in the mitochondria, which we have seen in the respiration process. Once the malate is formed, ultimately from the malate we'll get the sucrose. So what is the process of conversion of malate into sucrose? It's called as gluconeogenesis, and this occurs in case of cytosol. So this is the complete process of how lipid mobilization occurs. So starting with, if I want to recap, it is because of beta oxidation. After beta oxidation, it is glyoxylate cycle. The glyoxylate cycle, it is the partial citric acid cycle or partial Krebs cycle, and then at last it is because of gluconeogenesis. The sucrose is formed from triacyl glycerides or the lipids. This is just the overview of the mobilization of seed storage reserves. So starting with starch, proteins, and lipids. So this completes our seed physiology. I hope like I have covered all of the things which are necessary for your exam in case of floral biology and seed physiology. If you still feel that I have just skipped anything in between, you can just give me a text message in case of WhatsApp, or if you can tell me right now, you can just text in the chat box here. Otherwise, we can continue the next class and we will just start the legislation and the certification part. So from where you will get to see major type of major questions for your JRF exam. See, even if you get some questions in these particular aspects which we have discussed in this week or last week, so this is again overlapping with the content of either microbiology or the crop physiology, because since it is again the same respiration, photosynthesis, mobilization, development, so few things might overlap. So anyhow, whatever knowledge that you are gaining here, it will be ultimately very useful for you. So make sure to revise it from time to time. Okay. Anyhow, I will be providing you the notes. You need not to prepare it by yourself. So you just add, if you feel like anything is missed in this particular notes that I'm giving, just make a xerox of it and you can add it in this same book or same notes. Don't waste your time on making your own notes for this minor subjects. Why? Because for minor subjects you don't get to see the source which are self-sufficient in themselves. You need to again refer at least 3-4 books to understand a single topic, and that will consume so much of time. and that is again not even worth. Why? Because these questions are very basic, and questions are very, like, weightage is very less. You will get to see only around 10 to 12 questions from seed tech and 10 to 12 from microbiology. Yeah, yesterday's class will be uploaded by today. Now, immediately after the class, I will do it. I hope like the sessions are actually helping you. If you have any queries or if you have any suggestions of how we can improve ourselves or how we can just update ourselves so that people will understand these things better, you're always welcome. You can just text me in the WhatsApp, or you can just directly contact me anytime. Okay, whenever I am available, I will take your call. So if there are no doubts, we can meet in the next class. Okay, thanks for attending, and we'll meet in the next class. Good night.